Welcome, everyone. Welcome to the Meeting on the Mesa virtual edition. Happy to be here today to moderate a panel called Disruptive Technologies that will change the cell therapy industry. And delighted to be joined by a panel of renowned leaders in the sector, Dan, Helen, James, and Stefan. We all know that the industry is going through a pandemic, or the world is going through a pandemic, but really the cell and gene therapy industry is just booming at this point with record investments and deal making going on. And this is really because of the dynamics of innovation in the sector, not only in the recent years, but during this pandemic. And we're going to talk about that today and talk about how the disruption is happening across time and across different workflows and stages of the industry. So without further ado, I would like my panelists to introduce themselves and describe a little bit about the disruptive activities that are happening in their companies. So 3 to 5 minutes each. And Helen, why don't you start us off?
Thanks very much, and very happy to be here. I'd like to thank the organisers for having the opportunity to be part of a panel. My name is Helen Tayton-Martin. I'm the Chief Business Officer, but also a co-founder of Adaptimmune Therapeutics, a T-cell therapy focused company based on optimized affinity T-cell receptors focused in solid tumours. We've been around actually now since 2008 and on NASDAQ since 2015. So it's been quite a long journey to bring therapies forward. I'm a molecular immunologist by background and then worked in the pharmaceutical industry for various pharma companies and then in consultancy before becoming a co-founder of Adaptimmune. So I'm probably a lot older than I look, I hope. But anyway, and it's been pretty disruptive time, I have to say, through the entire period of Adaptimmune's history, pretty much from the very beginning, being involved in a gene and cell therapy company for that long at a time when cell therapies were really just academic endeavors, right through to where we are today, where they are mainstream therapies in certainly some indications in hematology. And we, like many, are focused on trying to bring these therapies to the mainstream in solid tumors, and in fact hope to be the first to bring a TCR T-cell therapy to market with a MAGE-A4 program, bringing that forward to launch in 2022. So exciting times. I mean, disruption, I think early days, as I say, just having a cell and gene therapy you could actually put into the clinic as a company, relied on collaborations, relied on willing clinicians, and patients, obviously, and really the ability to safely bring things forward was very much a challenge at the time. We're talking sort of 12 plus years ago. And I think the challenge continues as we think about bringing more disruptive therapies forward. And in my mind, that's, we'll come back onto this, is probably allogeneic or off-the-shelf approaches to try to get similar results. So I probably haven't taken up my full few minutes, but I think that probably gives an intro to me, to Adaptimmune, and to some of the disruption, past and present, that we probably would talk about.
Yeah, thanks, Helen. Thanks, welcome. Dan, why don't you go next?
Good morning. So I'm Dan Shoemaker. I'm the CSO at Therapeutics. I've been here coming up on 11 years, so I've really had a fun perspective to see the evolution that has happened to the cell therapy field. We're a 250-person company, a cell therapy company located in San Diego, and our focus is really developing iPS-derived, highly edited NK and T cell therapies. We believe strongly in universal off-the-shelf, products, and some of the advantages of iPSCs is it's just really a new manufacturing paradigm that uses these iPSC lines as a renewable starting material. And iPSC is an induced pluripotent stem cell, and there's two basic properties. One is unlimited self-renewal. So you could really think of it as a cell line, and we take advantage of that to sort of enable some complex gene editing strategies that I think would otherwise be very challenging when you're working with batches of terminally differentiated cells. And once you find your iPSC master cell line that has all the right edits, that then leads to the second advantage of iPSCs, which they can be differentiated into any of the 230 tissues in the body. At Fate, we really focused in on making hematopoietic cells and specifically NK and T cells. And so this is just an exciting time. Like if you go the last 2 years, back in 2018, we filed the first ever IND for an iPS-derived NK cell. And now fast forward to today, we have 9 cleared INDs that are in the clinic for both hematologic malignancies and solid tumors. And so it's really nice to see this first data emerging, showing the safety and feasibility of this new manufacturing paradigm, that you can take an iPS line and make NK cells and now T cells and put them into the clinic. And we're doing this in a multi-dose, multi-cycle fashion. So we're starting with non-engineered products, and now again we're at 9 cleared INDs. We now have products with 4 different edits, and it's an exciting time now as we're starting to see, in addition to the safety and feasibility, we're starting to see some evidence of clinical activity. And so I think this is the next 10 years as we really think about the ability to create scaled, off-the-shelf, highly edited cell therapies. I think this is going to be what it's going to take to win this battle against cancer. But it's still early innings. A lot of discoveries is left, but I think the progress in the whole field is, I could be more excited to be here.
Yeah, thanks, Dan. And in terms of COVID, I mean, has anything slowed down for you, or do you just see this as another step in the, kind of this kind of transformational, rapid expansion of the field?
For us, it's actually been nice in a funny way, because we're growing really fast right now, and we're growing out of our building. And so with the COVID, it sort of allowed folks like me who don't have to be in doing actual lab work to work remotely from home. And so a lot of the folks in regulatory and clinical development who don't have to be there are now working from home. So it's really allowed the people who have to be there to really take advantage of the footprint we have while we open up a new facility. But it's also nice to be able to sort of do the manufacturing ahead of time and have cryopreserved products that can be, shipped to your clinical site. So logistically, it makes everything a lot easier and we've been able to really continue the momentum of our enrollment and opening up new clinical sites. But that said, it's super challenging, and but we're, I think we're making do. There's some parts of working from home that I love, but I definitely miss the water cooler conversations and, even going to meetings like this. I think this is effective, but just that ability to really catch up with folks in between sessions. Those are the things I miss the most, but I've been pretty proud of our whole field, continuing to make progress in the context of such a challenging time.
Excellent. Thanks, Dan. James, why don't you go next? Welcome.
Thanks. So I'm James Trager. I'm Chief Scientific Officer at Nkarta. Have been working in cancer biology, cancer therapy for my entire career. Started out at Geron Corporation, part of the team there that cloned telomerase, and did some of the early work on embryonic stem cell biology, mostly as a near spectator and rather than a participant. Moved on from there to Dendreon, part of the team that got Provenge approved, so the first approved cellular immunotherapy for cancer. Spent about 13 years there, getting the approval and really getting the product to market and to some degree of commercial success. Had a very exciting and educational time there, but after 13 years, I stopped being scared going to work every day and decided it was time to move on. So took a position at Nkarta, joined there as the 3rd employee. We're now up above 80. Cleared our first IND a bit earlier this year. So getting ready to treat our first patient, which is, don't have to tell anybody here, incredibly exciting right now. Also went public earlier this year, which was gratifying. We're focused on primarily on the biology of natural killer cells, expanding them, engineering them, editing them for maximum efficacy in both hematological and eventually solid tumor settings. Again, really committed to the prospect of allogeneic off-the-shelf therapies. I mean, I think when when Provenge was approved, we kind of shocked the world with a $93,000 price tag, which in retrospect, like a lot of the technology, seems sort of quaint. But I think, the field really does need, for a variety of reasons, cost being one of them, access and availability, being really the main driver, I think needs to get to a more industrialized place, I would say, and off-the-shelf therapies, I think, really offer that. And natural killer cells, some of the other immune cell types, I think lend themselves to that very naturally and very easily. So that's our focus. I think made a lot of progress, and it's just an incredibly exciting time to be in this field right now.
Yeah, thanks, James. So, I mean, you've clearly been involved in a number of kind of what would be classified as disruptive eras or activities in the sector. Do you see the industry kind of coming together in the perspective that lots of different aspects of the industry are now going through disruption, as opposed to purely technological in the kind of the early days for you?
Oh, I do. I mean, I think you see it. I think all of us here probably understand what a huge team endeavor these things are, and a lot of this work was driven early on from the basic research of, our founder was Dario Campagna, Dan Kaufman and Jeff Miller and others in the NK field, the Carl Junes, the Michel Satellans and whatnot. But as time's gone on, we've seen this professionalization of the process development, the manufacturing technologies, the pharmacology around these products. And, with that, the sophistication and broad excitement on the part of the clinicians and treatment centers and the regulators. I think it's been, an ongoing journey for them to come up to speed on what these products look like, what to expect from regulatory filings for various cell therapies, what the right questions and touch points are. So it's, I think, the basic researchers are always the shock troops, but you really have to bring the army along to, I think, make progress in a field like this.
Great. Thank you. Stefan, welcome.
Hey, what great company. I know we talked a few weeks ago, but now that I listened to those introductions again, I'm in awe. Yes, Dan, we should have beers and not just sit virtually together. So my name is Stefan Wilt. I work for Takeda. My role at Takeda is twofold. I am head of pharmaceutical sciences, which is more of a classical CMC organization, but I'm also heading up a team that we call the Cell Therapy Translational Engine. And how did I end up in this field? Actually, I was in 2012, we're focusing on biologics, working for Novartis, when Novartis decided to place a huge bet on amazing results coming out of the University of Pennsylvania. Carl June was mentioned. And I stepped out of my back then role and was tasked to build a team to really figure out how one can do the non-clinical technical development piece around cell therapies. And as we all know, 8 years sounds like nothing, but in cell therapies, that is ancient. And that is how I got into this field. And talking about being disruptive, it was very intriguing to me to see how human cells could be used and engineered to acquire traits that we could use to hopefully, that was our aspiration, I still think is an aspiration, to be curative, which is ultimately, if we all can move beyond treating and we can cure, I think we will be very happy. That is a little bit the holy grail. So ever since I was very interested in cell therapies. And when I joined Takeda 3 years ago, Takeda was a complete newcomer in that space, in the sense of not having a strategic framework around various investments. So Takeda has a significant externalized R&D operating model with a lot of partnerships. And my role back then was to think with the team internally and see how Takeda could enter the cell therapy arena. We had already a very interesting, promising relationship with Yamanaka Sensei at T-Syra. He was awarded with the Nobel Prize in Medicines and Physiology for his work in inducible pluripotent stem cells. We had already began to talk to Adrian Hayday around a new class of T cells, gamma delta cells that are tissue residing. But we really didn't quite know how to put everything together. Now, fast forward 3 years later, we still have a lot of collaborations. We have an internal team that now sits downstream of discovery, has an integrated capability where it connects translational, correlative, technical, data, analytical sciences in one team to really work with partners to pull ideas forward into rapid clinical translation. And I'll just give you a few of those relationships that we have, and this also speaks to the uniqueness of our approach. I mentioned iPSC. I'm completely with Dan. If we can really engineer traits into a almost unlimited cell source, we are moving cell therapies much closer to where biologics are these days. Unit operations become easier, more mature, and then design is a huge feature. So lowering costs, access to medicine, as was mentioned, iPSCs have tremendous promise. I mentioned Adrian Hayday's work. Takeda invested in a build-to-buy company called Gamma Delta Therapeutics, where we're trying to more deeply understand the potential of these gamma delta cells, which we believe, as they are tissue residing, and they're somewhere between a classical alpha beta T cell and an innate T cell, that we think we have something that we can deploy towards solid tumors, for example. We have a deep relationship with technology coming out of Koji Tamada's lab, a company called Neulimmune Biotech, where we arm alpha beta T cells to overcome escape mechanisms of solid tumors. Again, moving beyond where the field already is, moving towards solid tumors, similar to what we're trying to do with gamma delta cells. We also have an amazing relationship with Michel Sadelain, who was mentioned, who really, and his team is just enormously creative on all fronts of cell therapies, as Dan probably knows firsthand, and just a beautiful person to collaborate with. And then lately we have now struck a relationship with MD Anderson, where Kati Rizvani's cord blood-derived NK cells will be tested for clinical indications. So our approach is varied. What's connected here is the idea to work in redirected immunity, exploiting as much as possible innate immunity. We can also, if Chris Arendt, my partner in crime, who's the head of the oncology therapeutic area unit at Takeda, would be here, he would also mention some of our T-cell engager programs, oncolytic virus programs, where we're trying, in the context of cold to hot and redirected immunity, use cell therapies, but also combine it potentially with other modalities. So that's in oncology. We're moving also into rare diseases, rare immunology diseases in GI, now that we have ex vivo gene therapy capabilities, but today we'll focus on oncology. Yeah. So, and it is a team sport. Very clear. While we compete on patients' behalf, but I'm a firm believer that we need to collaborate. Another COVID lesson. We need to really step back and ask ourselves, why are we here, and how can we get to where we want to best? Right. But we can talk about this also a little bit.
Yeah. Thanks, Stefan. And we're recording this, and I'm not sure I introduced myself.
What a miss! Yeah. You're not a selfish person. So,
yeah. I'm anyway. My name is Michael May. I'm the CEO of the CCRM, and was really proud to be a member of the board of ARM for the past 5 years. Have now moved on, but maybe we mentioned, or you all mentioned, or hinting at a number of kind of transformational approaches and developments. But let's think back to what, the last 10 years that ARM, the meeting on the Mesa, has actually existed. And let's just start to fire off some of the transformational technology or technological developments that have happened over that 5 to 7, 10-year period. So, who wants to start? What's happened in the last 10 years that's really changed the industry?
I'll give it a go.
Yeah, go ahead, Dan.
So it's been really neat watching the fields of cell therapy and gene editing sort of co-evolve. And And, in addition to the ability to, sequence genomes at very low cost, at very fast turnaround, and to see these two worlds, just the ability to apply gene editing tools to make cell therapy better. And I think the early work by Michelle Satterline really captures this the best, just putting in a chimeric antigen receptor into a T cell and going in and getting 70, 80, 90% complete responses in patients that had failed, all known therapies. And just to see that sort of the magic that happened when these two fields just came together. And really just showing the power of being able to, reprogram immune cells to fight cancer better. And as we now go from autologous and start thinking about allogeneic and off the shelf, I think that the fact that the gene editing toolbox continues to improve, the ability to knock genes out, insert genes, base modifiers, it's really, what we're doing in cell therapy could not be happening without that wonderful toolkit. And then I'll also say just the ability to do sort of single cell multi-omic analysis to really understand the properties that we're building into the cells themselves. And not only that, as we start bringing these into the clinic, the translational findings of really being able to understand the trafficking, persistence, how the adoptively transferred cells interact with the patient's immune system to really sort of orchestrate a cure, as Stefan was alluding to. I think that would be it. So if there's one toolbox or one innovation that has happened that I think has changed everything, it'd probably be the gene editing toolbox, and then what it's allowing us to do, and the Adaptimmune work that, really showing the ability to refine, enhance affinities of TCRs and just take immune cells and just put, more potency and maintain safety. And now as we go from understanding what's possible to then how do we actually scale and get, manufacturing is now becoming a core tenet of how do we now make these therapies that we're getting more and more confidence work. But now we just need to sort of figure out how do we make these broadly available to folks. So I would pick gene editing as my winning technology.
Yeah, well, you're hard pressed to maybe find a better one or a more clear one. But what else? Lots has happened.
Can I jump on top of that? Because I think,
go ahead.
In addition, you mentioned sort of our ability to characterize these cells on a single cell level through, we can look at sort of promoter usage. We can look at single cell RNA-seq. There's all kinds of technologies for really deeply characterizing these cells. And that's important translationally, as you mentioned. I think the other thing that really starts to enable is getting out of this paradigm, and I'm sure we've all heard it till we're sick, the process is the product, which I think has been harmful for this field. The product is the product. And we've been held back by the complexity of these products and in some ways the limitations of the tools that were applied. And primarily flow cytometry, which is, the greatest thing since sliced bread, but clearly it doesn't get at the heart of what's differentiating the cells in a lot of cases and how to best characterize the cells. And I think to continue to make progress on the manufacturing front, on the accessibility front, if you're locked into a process too quickly because that's how you've defined your product, it makes everything else extremely inflexible. So I think that characterization piece, again, it's extremely important from the point of view of understanding what you have and how it's going to interact with, with the patient's tumor, with their host immune system. But it's really important on the manufacturing side as well, I think, to get us to a point where we can expand these platforms.
So, if I may, can I jump in?
Go ahead,
on this also? Yeah. I'm actually, I think where James and Dan are going, the step function change in our ability to analyze and deconvolute and not just connect, as James was describing, the product design manufacturing piece with what happens in a patient, but also then developing feedback loops. And I think what drives this towards, and we may touch on this today, a rapid innovation cycle that now we can drive because we have improved analytical tools and we have improved data management tools. And we have, of course, a huge influx of ideas and innovation on the technical side, but also on the patient side, the understanding of what's going on in the patient. And then connecting those two is extremely powerful. That enormous acceleration that I've seen over the last 5 to 8 years is just enormous. I mean, it's not stopping. You know, we're approaching almost software or electronics types of innovation cycles here, and how to handle that will be an interesting challenge and opportunity for us.
Yeah, you know, what's interesting is that, you know, COVID has actually shown that we can work under even faster timelines because of the kind of decision by the regulatory agencies or government to actually come up with solutions faster. So I think we can go even faster, but it's those innovation cycle times are going down. Helen, how about you? Last 10 years?
Yeah. So the last 10 years, I think for me, a real red letter day was the very first actually the second sarcoma patient we treated with our NYESO program, an optimized TCR targeting NYESO in sarcoma, and the first complete response in a solid tumor we were working with the Crystal Mackles group at the National Cancer Institute at the time. The first time anyone at the NCI had ever seen a complete response in sarcoma. So I have that image on my wall, not here at home, but in the office. It sort of forms part of our history of the company as well, because it was such a humbling moment that a patient could actually, you could see a scan where every single tumor had gone, every single one, the CAT scan and the PET scan, sort of all of them side by side. And that happened within 3 months. So for me, that was a wow moment. This can really do something. And I agree with my fellow panelists. I think what we spend a lot of time doing with subsequent programs that we've brought forward under our own steam, and repeating that data actually in synovial sarcoma elsewhere with MAGE-A4, and then seeing responses with tumors disappearing and, you know, or shrinking and having real responses is what's driving that. Why do some patients respond and some don't, when you've got a pretty standard autologous cell process? So for us, having in-house capabilities, we can make cells in our own facility in Philadelphia, having translational capabilities either side of, we're in the south of Oxford in the UK and in Philadelphia. So we sort of iterate the samples and the translational findings. And so the power and the speed of that is becoming, more and more important, so that you can chase down those responses, so that you can actually get to ultimately a developable product that patients can afford, that payers can afford, and that then can become accessible. And I think where it really then links in is, what are the characteristics of a product that actually works in patients, and then how do you make and the off-the-shelf version? Because, you know, our translational teams on the autologous side are increasingly talking to our allogeneic team that are also working with iPSC-derived T cells to optimize the characteristics of the differentiation that we're planning for. So I think if we hadn't had, to be honest, if we hadn't had a complete response back in that time frame, we wouldn't have been able to do our original deal with GSK. We wouldn't have been able to finance the company. We wouldn't have been able to go forward. So it's then making good the belief that these cells can actually have a real profound impact on patients. And the last thing I will just say about it is that patient came into the company. And I have to tell you that there is nothing more powerful than having a patient who's benefited from the therapy come in and talk to the staff, who are excellent and super dedicated anyway. But when you've got sort of the researchers who made this stuff, and then the clinical teams and manufacturing teams who actually are able to sort of, they turn around patient material, sometimes against all the odds, when shipments we're shipping to Europe, we're shipping all over the US, and sometimes they just have to come in and work through the night to get those products out on time and released back to the patient. So making the manufacturing simpler, again, probably back to allogeneic, ultimately will make such a difference to rolling this out. But the reasons to believe are strong, and I think that's what keeps us all going. COVID or not, I think, we've carried on in the labs, we've carried on manufacturing cells and dosing patients, despite the odds, really. It's, I'm always so impressed with people's dedication in this field, not just in our company, but I'm sure in everyone else's too.
Thinking back 10 years ago, MSCs have had a bit of a resurgence in the COVID environment, but there was a real lack of efficacy 10 years ago. So I think you're referring to the products that are being developed are in the clinic and demonstrating that efficacy, which is driving investment and all that. Helen, do you think the clinical perspective, the clinical design, is that Is that changing? Are we more sophisticated in our clinical trials than we were 10 years ago, or is it just where the products are better?
Personally, I think that I like to think that we are evolving in clinical design to some extent. So I'll give you an example. We, with more recent autologous therapies, we used to have to start off at a very low dose from a safety perspective. I think in the early days, actually, you just made a rationale for a cell dose, and you went and actually, knowing that the T cell receptor wasn't going to have a toxicity effect on an off-target effect was, I think we learned that probably harder than anyone to sort of really understand what it is the cells could see. So that meant that you had to go through dose escalation, and that's one patient at a time. And I think that things like that can go faster. Instead of 3 patients, you could probably do that in 1, or even dose escalate within 1 patient, because ultimately clinicians really want to be able to put patients on trials where there's potentially some benefit. And if you're giving a sort of a homeopathic dose, but actually no cells, then the chances of benefit are remote. And in fact, we've actually gone back through dose escalation to a patient that was dosed earlier on, and redosed actually at the top dose, and so that we'll wait to see whether that has an effect. But we had the apheresis material stored, so we could do that. So I think there are some creative innovations in trial design. I think in the autologous space, getting access to apheresis material earlier, actually before patients have had too many other cycles of therapy, just from the work we do in the labs in working on the characteristics of the cell product, will probably make a difference. So that's a different model. How do you apherese before you've even, on diagnosis, before you've even tried anything else, which will probably come back to profiling of patients, understanding their markers, etc. So I think there has been some evolution. There is just so many new technologies out there, and so many companies trying to do new things. Some flexibility on that is important. Otherwise, it's too crowded. It's difficult to find patients, actually, because they've got so many options. You've got to think quite creatively about what's a good way to understand this. What's going to make sense for the patient and the clinician who you rely on to work with you to get patients through trials.
Nobody mentioned kind of the commoditization of reprogramming. The availability of the iPSC is. Can someone comment on that? I mean, that was 10 years ago. Was still in the early days.
Well, that's clearly Dan. Go.
Go ahead, Dan.
No, it's like I think it's just accessibility and having a reproducible, consistent starting material that could really take this whole cell therapy field more into how we think about manufacturing biologics, where you seed a bioreactor and make thousands to tens of thousands of doses. And that master cell bank that you use to initiate a run today will be the same one that you use, 2, 3, 5, 10 years from now. I think it's going to change everything just on the availability and reduce cost. And I think from cell therapy in the early days, the manufacturing complexity was so high that you really were, and complex, that you really were shooting for a one dose, one cure paradigm just because you had to. Not that necessarily that was in the best interest of the patient. And with our iPSC off-the-shelf platform is right out of the gate, we were able to do multi-dose, multi-cycle regimens where we could really think about, PK/PD profiles of how do we drive as deep and durable a response as possible over a 2-month window, where the idea of just having wave after wave after wave of fresh cells that are, consistently engineered is something we believe strongly in. It's been nice from an FDA perspective that they've had an open mind to really understand what we're trying to do, especially with NK cells that don't necessarily, and allogeneic NK cells that really are not meant to persist forever. But the idea that you can create that optimal 2-month experience of PK/PD really is, that door is open for you when you have a freezer filled with cost-effective doses. And so I think that's an example of how, having access to lots of a freezer filled with doses just changes how you think about clinical trial design and really doing what's right for the patients. And even on, as we think now about questions like NK versus T cells, my idea is that probably in the not too distant future, we'll be thinking about how do you combine adaptive and innate cells in a single dose. And maybe over a dose regimen, you'll start off with an innate heavy therapy to debulk a tumor, and then maybe switch more over to a mixture of NK and T cells, to sort of go clean up the last residual tumor. So it's just some fun things you can begin to imagine, but it all does come back to having a scalable, cost-effective manufacturing paradigm on the front end. And then I think it does open up some neat doors as we think about coming up with creative strategies to go for cures and not just treatments.
So does anyone have any comments on that? I mean, this idea of combination, combination therapy.
Yeah,
go ahead, Stefan.
I mean, I'm with Dan. I'm completely excited about this, and that's one of the main reasons why we built our strategy the way we did. And Dan, what you didn't mention, you could also completely see how you could combine NK cells, for example, with therapeutic antibodies, or some of the cold to hot strategies, soften the tumor, make it accessible for cell therapies, and then combine. But going back to where we all are going, the disruptive piece probably will be in deciphering how cells attack cancers and how cancers escape attacks, and then being able to understand that and go into an engineering space. I think that's back to where we were before. Analytics, readout, and then the ability to engineer your cell therapies towards desired traits. And even if I can, we're talking disruptive, if I can dream very long shot, maybe we can engineer these cells in situ. We don't need to make ex vivo gene therapy engineered cells anymore. Who knows? I don't know. I'm dreaming here. We're talking disruptive. And then we have deep understanding, and we simplify everything, and accessibility becomes really possible everywhere, not just in wealthy countries and highly or hospitals with sophisticated infrastructures, which is really something that irks me. I'd like to really make sure that this innovation that we're all part of becomes accessible to people and patients wherever they are. You should think about this.
Yeah. We're starting to transition to kind of future look. But Helen, do you have a response to that?
Yeah, I just have a challenge really for the other panelists, because I completely buy into the idea, having multiple cell types is probably going to be important. Having them accessible is going to be important. But how do you start the response in the first place, I think is crucial, because these are, if you take the autologous setting today, the cells that you take out of a patient and modify and give back, those cells were there to start with, and they weren't controlling the cancer. So in our case, we put an optimized T-cell receptor in because we know the patient has that target, and it lifts it up and can target it and sort of trigger a response. You put a second generation modifier in there too, and which we did with a CD8 alpha to make the CD4 cells just as potent as killers. So, but we had to have the targeting. We had to sort of have the sort of missile go in to sort of launch the inflammation. So I think there's definitely scope to use multiple cell types. It's how do you get the engagement, and then how do you ensure that it lasts and it's durable? And we've got some ideas within Adaptimmune with the second generation, and then ultimately that's what you want to have for your off-the-shelf stem cell to T-cell products. But I'd be interested, what do the others think about how do you get that response going in the first place? How do you light the fire?
James?
Yeah, no, and obviously coming from the Dendrion experience, I mean, our whole focus there was on using antigen presenting cells. So it was more on, we did have a target antigen, but ultimately it was about engaging the patient's immune system. And I think that's part of the promise of mixing the cell types, natural killers or gamma deltas. They have some ability to draw in DCs and other antigen presenting cells. Yeah, I think in solid tumors, it's certainly going to take a combination approach where you do need to manipulate the tumor microenvironment to some degree just to help the cells get in there. Obviously, manipulate the cells in order to help them survive. But I think, understanding this sort of dynamic between lymphodepletion and how that affects or hampers effective immune engagement is really key for the field. Because I do feel, we have sort of looked at these cells almost as surgical tools, and that can be effective. But I think, again, as we face the challenges in solid tumors, it's probably, we will be able to combine cell types, we will be able to combine with other modalities. But if you can't get the patient's immune system kind of re-engaged and exerting that kind of long-term control, I think it'll be difficult for, even some of the most powerful technologies to affect a durable, complete remissions.
I would add to that I think the key is being able to do multiplex editing, where things like dual targeting, building in cytokine supports, secreting factors that can overcome inhibitory factors of the tumor microenvironment. To me, these all fall into the category of must-haves. Like cancer just put up these barriers that just defeats the immune system. I mean, people have cancer in the midst of the immune system, and it's because cancer's figured out how to put enough barriers up that it blocks it. And the wonderful thing is, I think, as we now get translational data back and we start understanding these barriers, and then this is where you could take your gene editing toolkit and especially with an iPSC cell line approach, the ability to make products that have 3, 4, 5, 6 edits is feasible. And especially for solid tumors where you have trafficking, infiltration, recruitment, dual targeting, secretion of cytokines. And even now, I think these are like smart cell therapies that not only everything I just talked about, but then taking things that we know work like BiTEs, checkpoint inhibitors, and monoclonal antibodies. And rather than having to give those as separate biologics, the idea that these cells can be engineered to secrete these things in a local, very smart fashion just adds to the list of weapons that this living drug can deploy in a safe, effective way. And then again, having that cell go back into a truly scalable, cost-effective manufacturing paradigm that can be open that door back to multi-dosing, multi-cycle of cells that have, lots of tools to go to battle with the cancer against. That's why I think, back to the next question, probably like, what's the next 10 years look like? I think having platforms where we can really take these sort of lessons that we're each learning on the autologous CAR T therapy, what you guys have really revolutionized on understanding how to properly engineer TCRs to take something that wasn't quite getting there to getting there. And so now the idea is just taking these things and pulling them together and then focusing on accessibility and safety and really being able to deploy these types of solutions. And the type of edits that you'll put into an NK cell are probably going to be slightly different than what we do to T cells. But I do think this is where innovation is going to be key, moving forward. And it's early innings, but it's I think we know what needs to be done. It's just a matter of executing it and getting it done now.
Yeah, I mean, I think we've moved on to the future now. But thanks, Dan. But so all cell therapies in the future are going to be engineered or highly engineered. What do people think about a synthetic cell? I mean, are we going to get to a stage where we're not, we're working kind of from a baseline cell that can be engineered heavily, or just working with existing cells?
I mean, there are technologies emerging where red blood cells are being used to deliver nucleic acids, genes. It's happening as we speak. I don't know what will be truly successful, but I do think there are investments happening. There's innovation happening in that space.
Yeah. And at a certain point, I'm not sure what when does the android start and the robot start, I guess. I mean, I look at what Dan is doing, for instance, and these are synthetic cells in some senses, right? You're starting from a real precursor, but you can sculpt these things to such an incredible degree at this point that line between synthetic and real is a pretty fuzzy one in my mind.
Yeah, and oh, go ahead, Helen. No.
Yeah, no. I was just sort of build on that. I mean, that's very much the way we think about the iPSC-based T cells that sort of we're producing. Is that ultimately you'd get to a point where you'd be banking different second generation versions of those cells. So you could probably then drop in the right receptor for that patient. So ultimately, hopefully, you'd be taking a biopsy and you'd know which targeted receptor, whether it was a CAR or a TCR. And also you'd have some understanding of the profile of that patient's tumor. And so you might know which other second gen edits, which bank you're going to pull down to drop the TCR or CAR into. So I think to some extent it's already synthetic, because we had to work out all the biology to get to that point, so that you can bank something that's functional and consistent and off the shelf. So I think that's still looking away into the future, but the idea of customizing then what you give according to the profile of what the patient has, I think would probably be something that we'd all aim for, so that your chances of it being successful are much higher if it's customized, but you know what you're producing.
Dan, you raise a good point that, you know, we'll be engineering these cells, not just for their function, but for their manufacturability and for the analytics associated with them and all that. Any other comments on that? Before I want you to think about or give your opinion on gene editing. I mean, what are the bottlenecks there? I mean, this is a real driver of the industry. What has to happen in the gene editing space?
Well, James, do you want to go, or do you want to? I'll just give it a crack. So I think the gene editing toolbox is getting, you know, better almost every month. There's another Nature Science Cell paper showing that extending the capabilities. That said, when you do go beyond 2 edits to 3 to 4, no matter how good your editing tools are, when you're cutting the genome at 3 different places, there will be translocations, and there is heterogeneity, and you know, and especially this becomes especially apparent with the new, you know, 10x genomics type single cell analysis toolbox, where you could really look at the individual genomes of 10,000 individual cells and really understand when you go deploy these types of complex editing, the heterogeneity does become an issue. And again, one of the ways that we solve this is, again, we try and start off with the world's best editing toolbox to get as many cells in there properly edited. But the fact is that the majority of cells have translocations, copy number variation, off-target edits, and they're on a per cell basis, there is a high degree of heterogeneity, you know, and again, as you go into more complex editing, this becomes more of a problem. The nice thing about iPSCs is you can, you know, just do single cell cloning and then actually screen through hundreds of candidate clones and find ones that have pristine genomes, that have all the on-target edits and no unwanted off-target edits. We've also just introduced an idea where you could do iterative gene editing, where you start with a master cell bank that has 3 edits, and sort of what Helen was saying, you could just drop in a 4th edit to have a 4-point edited master cell bank. And I really think that's going to be, you know, the ability to do clone screening with cheap and effective genome-wide sequencing toolboxes, as well as the ability to do sort of iterative master cell bank generation. But it's challenging, and I think as good as the tools are, there's definitely need for better tools, less off-target activity, higher editing efficiencies, and again, the ability to insert genes, to do base editing, to knock genes out, it's really going to be these types of combination strategies that create these truly powerful, and especially in an allogeneic setting, to make these cells that are truly smart and can, you know, traffic and persist long enough and recruit the immune system and do multi-targeting. These are all the things that do require good editing tools. And we spend a lot of time thinking about this and trying to always get better at it.
That's well said. I mean, I think that there's we're starting to see technologies emerge that can help you to, especially in the case of genes where you might want to just alter their expression rather than make them a chimera, or, you know, alter their structure, where you can more globally imprint the cells without going in and, you know, cutting and pasting. And I think for rendering the, you know, the editing piece reliable and safe, you know, avoiding the edit when you can, you know, culturing the cells in certain ways, or using technology, you know, microRNAs or whatever it might be, to, you know, imprint the cell rather than actually edit. I think it's something, it's complex, and it's going to take, I think, a lot more just understanding of the basic biology, but I think that's promising to me in terms of really making a raft of changes that we'll probably need to make in order to get these cells to remain active in the tumor microenvironment.
Any other comments?
Yeah, just sort of one comment that I think the efficiency of the editing is obviously a major challenge, as Dan and James have talked about. The other thing for us, I'm sure Dan finds this too, is that where you put the edits can actually, you start with your stem cell and then you have to differentiate to your product cell. And where you put those edits, even if you think you've got them in the right place, can affect the biology. So what you're actually trying to produce can be affected. So you've got a sort of two-dimensional biology sort of challenge going on. One is actually where you put the edits and how you confirm that they're safe, and the other is the functional impact on the differentiation process of turning your stem cell, in our case, to a T cell. So you know, we've looked at lots of locations, for example, to put the TCR in to still get a functional T cell at the other end that can see the target and behave like an autologous cell. So that's just sort of one example. And then if you want to put second gens in that are also going to affect the T cell's biology, that could also affect the differentiation process. So there's I think what's the bottleneck? I think it's just biology, and it's hard, simply put. That's my view anyway.
Yeah, I would, you know, as I'm listening to this, clearly we're done with reading, we're starting to write, but the books are really not properly understood. So even if I think about an iPSC cell, even if we differentiate, to Helen's point, well, what do we not know, right? And then you start to add multiple edits to that. Being really sure that what we do is not just effective but also safe will be a continued challenge. How to do that right?
Let's spend a little time on the autologous versus allogeneic. What's the, you know, it's a you know, decades old debate, but what's going to have to happen to make those models successful?
Well, we happen to have both in our pipeline, and it turns one really into a schizophrenic person if you try to be successful in both. I wonder what Helen has to say here. Dan's clear; he's moved on. He wants to do all allogeneic, so we've lost him. But I think where Helen was going is there are intrinsic features to, for example, alpha beta T cells that we know work, and some of the challenges in terms of supply chains, logistics, manufacturing times, cost of goods. Enormous progress. So I would like to work backwards from clinical outcomes. So if something's really differentiated and curative, I think we will see continued autologous therapies. Even though I'm a huge fan of everything off the shelf, it gets simpler. The engineering examples, really understanding what to do better. So you can see I'm already schizophrenic. So I think there's space for both. But the little more appealing story clearly sits around the off-the-shelf allogeneic technology. But I'm, you know, these autologous therapies, they're powerful. I don't think we should just ignore them.
Yeah, I should jump in because I'm just as schizophrenic as Stefan here. You know, we have a autologous portfolio marching forwards to, as I mentioned, hopefully launching the first TCR-based autologous T-cell therapy. But we still, you know, and that sarcoma is a relatively small indication. We think we can do that at a commercial scale, and we think we'll have a product that can you know, reach globally. We can freeze it at both ends. So I think there is a lot on the supply side that you can solve for in terms of turnarounds, vein to vein time, logistics, tracking this, you know, tracking the cells, so the patient can see where their cells are, the clinician can see where their cells are. There's an awful lot that you can do to bring down cost of supply, and I think you can, you know, if you get the long-term benefit of a single dose of cells, that stacks up quite cost effectively actually for multiple dosing in some, you know, really quite toxic treatments over multiple cycles. So I think that there's definitely a case for the autologous products. The challenge is scaling them for very large indications, and that's where I think the off-the-shelf solution is, you know, absolutely where we want to get to. And so, you know, we've had a schizophrenic view of this for some time. We've been working on the allo programs for the last 4 years or so, 4.5 years. So I think, you know, ultimately they will get there, but I think there's a real place for the autologous products near term commercially. And it might take some time to see the same results with allo products. I think that's why it's IPS based, really. I think we're sort of believers on this panel in iPSC to get to T cells, NK cells, and probably other cell types as well eventually, but it's going to take some time.
I would agree. I mean, having spent a long time in the autologous field and jumped over, yeah, I certainly see the advantages of autologous, and again, the potential for one dose to be enough. Just the obvious marriage between the product and the patient is important. And, on costs, I guess when I was at Dendreon, one of my basic homilies was that, the first course in chemical engineering was one taught at MIT and one in Britain around 1865. And it's not like people knew right away how to make 10,000 liters of ethyl acetate cost effectively. Yeah. And I think we've seen progressively that the costs in this field have come down as there's more focus on the space and more suppliers of even the trivial things like bags and tubing can really have profound effects on your costs. And I don't doubt that we'll be able to drive costs down in the autologous field. Probably not as far as in the
James. We have already. Yeah. Sorry for interrupting you, but exactly. it's been enormous. I mean, what has happened?
Yeah. No, and even at Dendreon, I mean, it was, we went down an order of magnitude in costs over the course of 10 years or so. I mean, you just have to focus on it. I think you'll always have the issue, especially with later stage, cancer patients, that, there may just be issues of consistency in manufacturing from that patient's apheresis. And that's why the idea of taking the apher later on in the process is very attractive. And as for any therapy, moving it earlier in the treatment paradigm is, I think, pretty critical.
I'm biased, I'm an engineer, but the engineers always figure it out at some point, right? So Dan, what about...
Completely unbiased comment, yeah.
Yeah. Dan, in terms of immune cloaking, how far are we from that in terms of engineering, do you think?
So this is a great question, and it's a fascinating area of research right now in the field of cell therapy. The approach that we took at Fate is we really wanted to understand the problem before we started deploying solutions. And preclinically, we've been working on a lot of different stealth solutions that we could talk about in a second. But first and foremost is we really don't know from an example of a T cell versus an NK cell, are they equally immunogenic? And so starting off with our first product, FT500, which is a non-genetically engineered iPS-derived NK cell, we administered 6 doses over a 45-day period. And we really did not know if dose 4, 5, and 6 would be immediately recognized and eliminated by either a T or B cell anti-product program. And what we're seeing, at least initially, is that our iPS-derived NK cells are not kicking off these anti-product programs, at least within this first 45-day window, with a flu-cy conditioning regimen. And so I think it's such a neat system with conditioning regimen, and then the immunogenicity of the cell that you're putting in. We just need it to persist long enough to do the job, and then almost by definition go away. It's almost a part of the feature of the product as the patient's immune system comes back. That said, there's some wonderful gene editing strategies. We just licensed in this new technology called alloimmune defense receptors from Baylor, and basically what it does is it's a chimeric receptor with a 4-1BB ligand that basically recognizes and kills highly expressing 4-1BB alloreactive T cells that come in to kill your product. Then they get killed, and so you basically eliminate a subset of T cells that is the anti-product component. And so preclinically, we're exploring some sophisticated strategies that don't just wipe out the whole immune system, because I do believe ultimately what we're trying to do is re-engage the patient's immune system to truly get these durable cures. And so I'm a little skeptical on approaches that just completely wipe out the entire immune system as a strategy. And so we're trying to understand, with a outpatient conditioning and a multi-dose cycle, what is the problem we're solving, and then try and be as gentle as possible on the conditioning to preserve as much as possible the patient's immune system. But at the same time, you need your cells to persist long enough to do their job. And again, we're huge believers in multi-dosing, and so you have to make sure your last dose is as effective as the first dose. And so I just think it's an interesting time. And back to the allo versus auto question, I think it takes a village. I mean, the early work in autologous CAR T therapy was, revolutionary. The idea that you could genetically program immune cells to fight cancer and see those types of amazing clinical results, the ability to engineer in TCRs and to cure uncurable cancers. Like without those results, nothing else would be possible. And then the whole wave of allogeneic cell therapy, donor-derived, both T cell and NK cell, really ushered in the ideas of what are the rules of engagement to effectively safely administer a allogeneic cell therapy. And again, if that didn't work, what we're doing with iPS-derived off-the-shelf versions doesn't even make sense. So it's really, we're standing on the shoulders of a whole community. And then I think now we're at a perspective to truly say, what from 2 points is like, how do we take cost of goods down lower, and how do you make hundreds of thousands of doses, practically speaking? And the thing I get most excited about the iPSC platform is that is definitely a piece of it. But I also think as we understand the multiple barriers that cancers put up to avoid immune attacks, I just think it's going to require highly edited cells to overcome those very known barriers. And so having a manufacturing platform that can also incorporate complex editing strategies, I think I would put that at the top of the list as to why it's worth figuring out all the complexities of creating an iPS-derived cell therapy. And there's going to be a lot of great things that happen with the sort of generation 2, products where we are today. But I think especially as we look towards solid tumors, part of a long-term solution, which again is why a lot of the folks on this call here are all investing in iPSCs as a long-term solution, is I think ultimately it is going to take that type of highly edited approach, and then ideally that's going to fit into a scalable manufacturing paradigm. So long answer to a short question, but yeah.
Stefan, when we talked previously, you talked about some adjacent kind of disruptions that are happening across the sector. Maybe you can talk about some of that'll impact cell and gene therapy.
Jog my memory. Where was I going?
Like being able to manage data and software and stuff like that.
Yeah, really. I mean, that to me, I sometimes like to step back and look for non-obvious things. And so I have friends in software and electronics industry, but I also have a friend in the fashion industry. And I describe to them what we're doing in terms of trying to safely deliver sophisticated therapies, but while we do that, also continue to innovate. And there clearly are feedback loops that exist outside of healthcare, where we can look and we can try to get inspired. And I think it's not a coincidence that now we see quite a few silicon investors moving into healthcare, because clearly we're for once we're dealing with enormous amounts of information. You listen to Dan. I mean, Dan could go on all night. And why is that? Because there's so much information in the patient, in a hospital setting, in his labs, in Helen's labs, in James' labs, in the Takeda labs, and we're just only at the beginning. And into this amazing data space, we're developing methodologies to understand that information, and we need to also be able to deconvolute. I mean, Dan speaks about multiplexing, several edits in iPSC types. If I were to challenge him, he doesn't even know whether they're truly differentiated or not, or what he's not seeing. Then that we introduce into a highly variable patient population in many different hospitals, in many different countries across the globe, in different regulatory environments. So it's very clear to me that we need to take a step back and really think about how we handle information, how we handle data, how we mine information, and also engage with our partners. And partners are hospitals, regulators, we as an industry. So to me, this analytics data piece is something I always felt is not talked about enough, because that is where true differentiation will happen. And you look just what's going on, AI. I mean, we're getting as a society, as humans, so sophisticated in dealing with information. We need to pull this into our business more aggressively. I think there's a lot of potential.
Yeah.
Right.
Yeah, amazing. So look, we have to close off. I feel like actually we're just getting warmed up, and that we could talk for hours, but we need to close off the session. Maybe just quickly look into the future 10 years, and name one or two things that you think will be hallmarks of that future in the field.
I need to go back to where Helen was. You know, it's aren't we all here to help to cure cancer? We will have made a difference. We have to, and that's what excites us. So if I look 10 years ahead, and even just a little bit of that promise that we believe in has been translated into outcome for patients, it's cool stuff.
Yeah. Helen, what do you think?
Yeah, no, I agree with that. And I think what will help us get there is the sort of step change in analytics on the patient. So we'll be profiling patients with much greater understanding, and then the products we will be giving them will be much more customized, whether it's the targeting moiety, the second gen that we put in there, and hopefully that will be in an off-the-shelf version of that cell, so that it's, you know, it's accessible more broadly and cost effectively. So I think that cell therapy is here to stay, and I feel very, you know, privileged to sort of live through a sort of decade or more of that beginning to become something that is, you know, a little bit more well known in the healthcare field more broadly.
Dan, what about you?
I'm excited to see solid tumors fall to cell therapy, and just the fact that it's such a challenging, you know, it's where 90% of the patients are. There's such huge unmet need. And I do think having, you know, living drugs, smart therapies that bring multiple layers of anti-cancer activity that can be, you know, it's really going to be what it's going to take to overcome solid tumors. But I would love to see cancer just be eradicated in the next decade. And I think cell therapy is going to lead that charge, coupled with, you know, all the other advances from, you know, bispecifics and antibodies and checkpoint. Like, I just think that the progress in the field is making is phenomenal, but I do look to see cell therapy take a lead on the innovation, just because of the power of having a living drug that can deploy multiple levels of therapeutic activity, and you know, and then ideally be cost effectively manufactured to where we really can get these badly needed solutions to patients all over the world. So that would be my dream there.
All right, James, beat that. What's the future?
Yeah, I mean, when I look, I'll try. You know, so when, I guess I'm old enough, Stefan is probably old enough. I mean, I think when I look at the revolution that's happened in science since I first went to grad school, I would characterize it as really, you know, a breaking down of the barriers between all the disciplines. I mean, when I first started grad school, there was a genetics department and a microbiology department and a biochemistry department. And I'm sure you can still find that, but in a lot of ways, those distinctions have just melted. And, you know, when we look at cell therapy, in some ways, it's sort of the culmination of bringing together the immunology and the cell biology, the genetics, molecular biology, informatics. I look at what we haven't picked up yet. I think we still don't have great pharmacology models. And I think that impacts a lot on how we do the translation and how we really, you know, turn the crank on developing new products and understanding how they're going to work. But I think, you know, we're going to continue to see this sort of melting of barriers and assimilation of technologies and of biology. I mean, I think some of the problems that you guys are probably thinking about more on a day-to-day basis around cell metabolism and, you know, glycolysis that you haven't had to think about. I haven't had to think about anyway in the last 30 years. What's going on in pregnancy that, you know, to allow kind of immune toleration, these, you know, very basic scientific questions that all used to be different groups, different departments, different schools. I think we're going to continue to see them kind of just blend together.
And do you think that fusion of all of those skills will define leaders of the future as well, whether they're technical leaders or business leaders?
I think it is already.
Yeah.
We look for so-called multilingual scientists. It's the only way, and that's not just among the disciplines that James is also explaining. It's also in terms of research to development to commercial. You need to have that agility and be multilingual, which is really part of the excitement. I mean, I don't know whether any grad students or students are listening. This idea of having a great community to come together from different fields and make a difference, it's just inspiring.
Well, you know what?
James is right.
I'm going to end there with and show you a gift that my kids gave me. which might reflect a little bit on the way they look at the world. But maybe I'll leave that there. And
there you go.
Hopefully that's not censored, but I think it reflects what we're all talking about. I want to thank each and every one of you for participating today. Like I said, I think we could go on for hours. I hope everyone enjoys this session and enjoys the meeting on the Mesa in general. So thanks very much, everyone.
Thank you.
Thank you.
Thank you, Michael.
It's a pleasure talking to you guys. Stay well. Bye.
Thanks a lot.